Hold on, I have to show you this great thing that David Ryan sent me last night.
Oh, was it the little birds dancing? Yeah, I also got it too, and I was like, this is life. And I watched all of them.
I watched it more than one time. I was just like I was like, just rocking out, having a great night.
The one, I think it was the third or the fourth one was the best. It was, I forgot the song, but it was just so funny. I was laughing on my bed. It's just beautiful. Cute cut tomatoes. Everyone's working with tomatoes.
Well, they are a model.
They are, including me. Actually, no, I'm more tobacco. But not the smoking tobacco.
Oh, I didn't know the one was tobacco.
Yeah, different. Nicotiana tobacco is the one you smoke. I'm doing Benthamiana, which is like, it still smells like tobacco, so it's like, hey. So she's in the rose. I just had a coffee, so I'm really like.
I already had one coffee. I'm pretty sensitive to caffeine, so I don't drink it very much.
Yeah, same.
Literally, the only caffeine I regularly drink is grapes.
Mm-hmm.
Coffee is like I have to study or I have to stay alive type of thing.
Have you ever had the Awake bars, like the chocolate?
I haven't, but I've heard of them.
So they're all over campus because like people at Cornell, they like survive off of them. And I've never had one. My sister eats like 3 of them. Each one is like 2.5 cups of coffee. So she's just like. But yeah, I'm excited to try one eventually.
We don't want to start them until finally.
No, see, I can't do that because then I won't. If I have too much caffeine, trying to focus on studying and stuff. Because I don't really get tired that easily.
I don't know why I do know why. I kind of went to bed late. It makes sense why I'm exhausted right now.
If I drink, then I get exhausted. I'm just like.
I'll be so bad. But no regrets.
Oh, I have to pack up my stuff today. I'm leaving tomorrow. I'm working until 5:30 here, and then I'm gonna leave after that. My grandfather's coming. It is, but this car is quite tiny, so I have to like get everything in there. I brought way too much stuff. I think I wore about 3 outfits from this one. I think I brought 30 outfits, and I wore like 3 of them.
My problem was mostly like Delaney's clothes were like dresses and shorts and lab.
Really, yeah.
There's so much question. I brought a bunch of clothes and like only one bag.
I had like a few pairs of pants and I just like shuffled through them. Admiring your stickers. I have a few on.
So sorry. This one I'm sitting here.
phone is on. My friend. This is my friend's emoji. He also has the ring.
Just like any like. I mean,
I'd like to introduce our next speaker, Eleanore Ritter, who's been working in Josh Rose's lab and working with Mike Sigerson, and will be telling us about tomato kidneys. And you know how to use the demo.
So thank you, George, for the introduction. As he said, my name is Eleanore Ritter. I'm from Skidmore College. However, I spent the summer at the Rose Lab on my project, characterization of a putative cutinate from tomato fruits. Those of you that know the Rose Lab know that a big focus is on the cuticle. Approximately 450 million years ago, photosynthetic organisms colonized land. And as they moved from water onto land, they were subjected to increased environmental stressors, such as UV stress, water loss, and they really needed a way to mitigate these new stressors, and the cuticle likely developed to do so. It provides protection against water loss, UV stress, pathogens. It also prevents organ fusion. So overall, it's overwhelmingly important for plants, and it's composed primarily of cutin that's covered in waxes. So you can see there's a wax film on top of 2 layers of cutin that then cover the polysaccharide cell wall of epidermal cells. And so it's produced exclusively by epidermal cells. And a major component of it, as I said earlier, is cutin, which is a polymer. Cutin is a polyester primarily composed of polyhydroxylated transester fatty acids. And the monomer composition is well known, with a glycerol, which I'll be representing as a ball throughout this talk, an esteron, which you can see in purple, and a 16-carbon fatty acid chain. And while we know a fair amount about the monomer, we still don't know to what extent free glycerol is incorporated into the cuticle, as well as how the polymer structure is created, broken down, whether it's linear or branched. There's still a lot of unknowns. However, our lab was instrumental in identifying cutin synthase in tomato fruits, SL meaning tomato. And our lab found that cutin synthase catalyzes polymerization of cutin monomers, and it's required for polymerization of 90% of cutin. And this protein acts by basically acting on these cutin monomers, which I mentioned earlier, what we know of in tomato. And Cus1 will cleave the glycerol and form the ester bond between the 2 fatty acid chains. And this protein is within the GDSL superfamily of proteins in a subfamily of cutin synthase proteins. The GDSL family is composed of esterases and lipases that have a wide substrate array, however, also have a GDSL amino acid portion conserved in the catalytic portion of the protein. And you can see a very truncated version of the family here with only tomato and Arabidopsis present. And Cus1, as I mentioned, is in the cutin synthase-like protein family. However, a paper recently looked into ATCDF, or Arabidopsis cutin destructing factor, which thought to be a cutinase, or an enzyme that degrades cutin, based on the name. You can kind of guess that. And a TEM showed that when it was ectopically expressed, basically it was expressed where it was typically not found, and caused cuticle deformities such as peeling, which you can see in the black box, as well as absent cuticle, which you can see in the white box. And my project focuses on SL CDEF, which is not the cutin synthase-like protein family, and its function is basically unknown. And it's the tomato ortholog of the cutin disrupting factor that I previously introduced. And it appears to be a cuticle-associated gene for a few reasons. So on the right, you can see a heat map of the pericarp, of a cross-section of the pericarp of tomato fruit. And basically, the darker color is higher expression, which in this case is orange. And so you can see it's mostly expressed in the epidermis, and its highest expression is at 10 days post-synthesis, which is when the fruit is undergoing the most expansion, and there's the most cutin biosynthesis at this point. So, given these factors, we think that it could be a cuticle-associated gene. However, the function is unknown, and so it could be a cutin synthase, it could be a hydrolase. However, my project is to characterize this protein. So we took two routes to do so, the first being the in vitro route. So we purified transiently expressed CDEF protein in tobacco that was expressed with a His tag so that we could then purify it using negatively charged resin. And using our purified protein, we then determined the pH optimum using a free glycerol assay. And we also tested its cutinase function using a Molotov mass spec. For the free glycerol assay, we basically incubated our protein with the monomer, and given that it's a GDSL protein, expected it to break this ester bond. So the glycerol would be released, and then it would react with the free glycerol reagent through a series of enzymatic steps, which would cause the reagent to turn a purple color that could then be measured at 560 nanometers. So what we found was an optimum pH at 5, which is about the extracellular pH where the cuticle is synthesized. And it also matches previous findings of the optimal pH of CUS1. And so knowing this, we were able to gauge future experiments of what pH would be best for a protein's function. And it also gave us another idea that it could be cuticle associated. So moving into our Molotov experiments, basically, first we had to set up protein incubation, and so I'm going to go through acyltransferase activity, which is on the left first. Basically, the protein, and in this case it's CUS1, and this is how we think CUS1 functions, will cleave the glycerol to form an ester bond between the fatty acid chains, and this is simplified, it can form larger polymers. However, for esterase activity, we would expect that it could cleave an ester bond between two fatty acid chains, or it could cleave an ester bond between the glycerol and the fatty acid chain, or it could do both. And so once we have these reactions run, we can run them through a Molotov. And so you can see the polymers are present in increasing size. And the spectrum will give us intensity versus mass charge. So you can see the polymers are increasing in size as they go across the x-axis. And we know the molecular weights of the polymers, so we're then able to see if spectra are present when we run these experiments. However, with a potential cutinase, what we would expect to see is potentially the hydrolysis product of the polymers. So you can see that there's no glycerol present. And when we run it, we would just expect to see them shifted over without the glycerol on our spectra. And what we found for our boiled spectra was a peak at 385, which coincides with the monomer. And for CUS1, what we found was the polymers present. And note the absence of a monomer peak at this point. CUS1 was able to use up all of the monomers in the production of the polymers. And for CDEF, what we found was the hydrolysis product of the monomer, as well as the dimer. And the dimer can naturally form as it's incubated, so seeing the hydrolysis product of this is not terribly surprising. And when we combine the two, this experiment was basically run by letting CUS1 first run overnight and use up all the monomer, and then incubate with our CDEF protein. And what we found was, once again, the polymers were present. However, this time we saw hydrolysis product of the monomer, the dimer, and the trimer. And the peak at 311 is the most exciting. So at this point, the monomer would have been used up by +1. However, CDEF appears to be breaking not only the ester bond between the glycerol and the fatty acid chain, but potentially the bond between two fatty acid chains. And we then moved into our in vivo experiment with our generated CRISPR lines that we then used to image the cuticle using oil redo staining. And we also isolated the cuticle and analyzed it using gas chromatography to look at the biochemistry. For CRISPR plants, there was a 6 base pair deletion, which you can see in the RNA target, which basically caused shifting and ultimately changed 3 amino acids, including the L in the GDSL amino acid sequence, which I explained earlier is catalytically important for these proteins. And it also resulted in 2 amino acid deletions. And this was a heterozygous mutation that ultimately caused the phenotype, which is not too easy to see now, but if you look closer, you can see there are these ridges of hardened cuticle present on the outside of our fruits, as well as cracking. And both of these phenotypes were fairly universal on pretty much all of the fruits. And when we looked closer, using oil redo staining by basically peeling off the cuticle and staining it with oil redo stain, we once again saw a difference. So in M82 or wild-type plants, you can see a fairly uniform cuticle at both 20 days post-anthesis and in mature green fruits. However, in our CRISPR plants, you can see these protrusions of cuticle present, as well as some staining beneath epidermal cells, which is not found in mature green fruits. And we then measured the cuticle thickness, which you can see in this little bracket here, and once again found that there was a significantly thicker cuticle in our CDEF plants than in the M82 plants at both 20 days post-synthesis and in mature green fruits. Just to confirm this further, we use gas chromatography by basically peeling off the cuticle, breaking down the polymers using a strong base, labeling the monomers with TMS, and then running it with solvent and standards through the gas chromatography instrument. We once again saw increased cuticle coverage in our CDEF CRISPR plants. And so this confirmed what we saw in oil redo staining. And for all of my results, basically in vitro CDEF has esterase activity. However, we still don't know the optimal substrate for it in vivo. And cuticle deformities did appear to result from a mutation in the CDEF gene, such as uneven deposition as well as excess cutin present. And so CDEF could play a role in removing excess cutin, especially during fruit expansion. And it is possible that structural modification may be required to make a mature cuticle, given when it's expressed. And for future work, what we hope to do is generate recombinant mutant CDEF based on our CRISPR mutation to then test its esterase activity in vitro to see if it is reduced, which we would expect. And we also hope to study the cuticle ultrastructure, such as previous studies did using electron microscopy. And lastly, I would just like to thank the Rose Lab, my PI, Dr. Rose, as well as my mentor, Nick Sigerson, and the rest of the lab for all their support this summer. I would also like to thank our collaborators at DTU, and last but not least, BTI, George Shander and Delaney Sickler for all their support this summer and their dedication to undergraduate science, and lastly, the NSF for funding.
Time for a few questions. Any questions?
So, what does it mean to say that proteins are coming from the same family? Are they coming from some sort of a protein precursor that eventually branches out into all these various proteins, or is this more like an evolutionary thing?
Well, basically, they appear to be related, and I think the GDSL sequence is the one most important for this family since it's conserved. And there are different branches. You can go back. But I think that's the most important part with respect to the family that I was working with, was that they all have the GDSL amino acid sequence. Any other questions?
I guess we can move on to the next slide. So our next speaker is Carla Dizon, who's in Wojciech lab, and she's working with Lisa Olson right there. And so Carla will be telling us about polyploidy and meiosis.
Good morning, everyone. Thank you for the introduction. This summer, I had the opportunity to work in a Pulaski laboratory studying the impact of polyploidization on meiotic recombination. Now, first of all, what are polyploids? Polyploids are organisms or cells that contain multiple sets of chromosomes. Throughout the evolutionary history of plants, as you can figure, multiple polyploidy events have occurred, and many studies suggest that these polyploidy events have contributed to providing genetic material for plants to survive in challenging environments. In addition to being able to withstand living in extreme conditions, polyploidy is also selected for in agricultural crops such as strawberries, bananas, and watermelon to produce bigger and seedless fruits. And about 30 to 70% of the eggs and angiosperms are polyploids. Although it may seem that polyploid is beneficial to plants, many face challenges. One of the great challenges that they face are issues in meiosis. Now, meiosis is a cell division process which creates a genetic variation and ultimately leads to the creation of gametes or spores. Because this process is essential in the reproduction and creating genetic variation, polyploids must overcome these challenges in order to become successful. Now, by looking at the mechanisms of meiosis of polyploids, we're able to uncover specific difficulties that they face. In many meiotic research, maize is a widely used plant model because the meiosis in maize is synchronized. As you can see in the figure, the red circle indicates the center of the tassel, which is the male reproductive part of maize. Now, if you were to take anthers going from the center to the top or center to the bottom and determine the meiotic stages, you will observe that the process goes from the later stages to the earlier stages. So it's very easy to determine the meiotic stages just by looking at the location of the tassel, the anthers on the tassel. So in my study, I'll be using synthetic maize polyploids and their diploid progenitors. There are two types of polyploids: the allopolyploids, which are created using two different parental species, and autopolyploids, which are created using the same parental species. Due to the large genetic variation in maize, I will be using autopolyploids. And because of this genetic variation, the autopolyploids will be divided into two different groups: the homozygous, which contain the OHI43 and B73 line, and the heterozygous, which is a cross between the two. Okay, so the first part of my project is to determine if polyploidization has an effect on the later stages of meiotic recombination. So in meiosis, the main focus of this would be prophase 1, which is the longest and vital process in meiosis, and is divided into 5 different substages. Within this stage, I'll be looking at two link processes, which are recombination and synapsis. Recombination begins during the leptotene stage, where double-strand breaks are formed. The first figure, and the second figure is followed by reception, which creates the single-strand overhangs. And during zygotene, these single-strand overhangs invade the double-strand pieces of DNA. Now, in the previous studies that were done in the lab on these early recombination stages, the double-strand breaks of polyploids were much greater than the diploids. And looking into the bacotene stage, where these double-strand breaks are expected to be resolved, they persist in the polyploids, which suggests that there may be a delay or a disruption in the resolution of these double-strand breaks, and may cause setback in the later stages of recombination, and this is where my project comes in. So after single-end invasion, the recombination intermediates then move on to three different crossover pathways, they can be resolved as non-crossovers, class 1 crossovers, or class 2 crossovers. Because 90% of the final meiotic crossovers are class 1, that's what my project will be focusing on. As you can see, there's multiple proteins that are associated with the class 1 crossovers, but one protein in particular, the focus of this project, is called K10, and it has been found in the early to late stages of recombination and is essential to the formation of class 1 crossovers. So for that reason, K10 will be used as a marker for crossover sites. Now, looking at the second process that will be focused on my project, synapsis, which occurs during the zygotene and cacotene stage. It involves the formation of a proteinaceous structure called the synaptonemal complex. And within this synaptonemal complex, there's a zipper-like structure that is formed by a protein called ZIP1. Now, many studies have been done on this synaptonemal complex, and they've demonstrated that the whole process of synapsis is dependent upon ZIP1. And in the absence of ZIP1, the whole stage of prophase 1 is delayed and non-homologous chromosomes form. So ZIP1 is also essential in ensuring that there's fidelity of chromosome association. Okay. Now, in order to study to determine the polyploidization effects on crossovers, I have done co-immuno localization of the two proteins, ZIP1 to mark synapsis and K10 to mark crossovers on polyacrylamide pads. And in order to quantify these crossovers, I've used two different types of microscopy, which is the wide field fluorescence microscopy and the super resolution structured illumination microscopy, or SRSIM. I will be going over the first one, which is the wide field fluorescence microscopy. The immunolocalization protocol for this is very long. It took 5 days. It's basically using a technique in which the illumination is used on the entire field view of fluorescence microscopes. Now, this technique actually picks up a lot of the blurred background noise of the sample. So an algorithm called deconvolution is used to reassign this blurred light back to its source to produce a better image. Going over the second one, which is SRSIM, the protocol for this was just 2 days because we use an enzyme cocktail to separate the cytoplasm from the nuclei, which makes it easier to look at our chromosomes. This has not been done before to detect the K10 protein, so we wanted to see if we're able to. It's based on the Moray effect, which involves the formation of a grid pattern light, which is caused by the interference of the diffraction grating. And this red pattern is then superimposed on the sample and is rotated 3 times in between taking the images. And the specialized algorithm then produces a reconstructed image, which it has a very good resolution, as you can see. In our examples, the wide field is very blurry, and wide field with deconvolution is a bit better, but the SR sim produces the highest quality image. Now, we've had technical difficulties with our wide field fluorescence microscope, so we weren't able to obtain images from those samples, but we were able to look at them using the SR sim. And as you can see in our B73 diploids and polyploids, there is ZIP1, which suggests that synapsis is occurring, and that is just to stain the chromosome, but there was no presence of HATE10. Like I said before, this was our first time using this protocol, so this wasn't optimized to detect the HATE10 protein. Now, because we weren't able to get the quantification of crossovers, we went on to try to determine if polyploidization has an effect on the results of meiosis. And we did this by counting the number of viable gametes of female meiotic product, which is pollen. In order to determine the viability, we've used the Alexander stain solution, which contains 2 different dyes, the malachite green, which stains the pollen walls, and the acid pyxin, which stains the mitochondria, cytoplasm, basically the protoplasm of the pollen. And this is how we determine it. The non-viable is green and the dark purple is viable. And these are the results. The plants were, when comparing the diploids with our heterozygous polyploid, there is no significant difference. And unfortunately, we weren't able to grow much of our homozygous polyploid, but we did manage to get one B73 polyploid to grow. And I wasn't able to add it into the previous results because of this peculiar looking pollen. We weren't able to determine whether they were viable or not. So we created an intermediate level. And here are the results for the B73 polyploid. More than 50% are non-viable, and 20 are viable, and the rest are the undetermined. So, just to wrap this up, although there were some issues in the earlier recombination stages in polyploids, based on our pollen viability results for the heterozygous polyploid, it seems that there may have been some resolution in the later stages of recombination to resolve the issues. And throughout the whole summer, we've had difficulties growing our homozygous polyploids. So there might be issues there. We need to wait for the plants to grow. But looking at just by the B73 polyploids, you can see that maybe the homozygous polyploids are having more issues than the heterozygous polyploids. And yeah, I'd like to thank my lab, my amazing mentor, and my PI, who was very patient with all of my questions, and Adele, who was there for my questions when the first 2 weren't there. And the BTI, George Gander, and Delaney for being such great coordinators, and the NSF for funding. Okay. Any questions?
I was wondering, why is it that polyploids generally are more successful than other plants? Like what does the polyploid individual plants that makes them more successful?
Well, by having multiple sets of chromosomes, there's a possibility of having more genetic variation, because that's one of the main reasons why they stated that they had a better adaptation to surviving in challenging environments because of the genetic variation that the polyploid gives them.
Yeah, Carla.
Oh, hi.
Yeah, thanks, that was great. So I'm just, I think I missed it. Those are synthetic polyploids?
Yes.
Okay, and then, so, you know, from work that you've been around or in literature, is there evidence that some of the early disruptions in quote-unquote younger polyploids are sort of less of an issue in older polyploids? So non-synthetic ones, I guess, things that have been around for.
Are you talking about non-synthetic polyploid?
Sure, yeah.
Polyploid maize do not grow in the wild, right? That's why they're synthetic. But we were trying to see if the issues in meiosis have caused them to adapt and for them to become diploids. Because the maize had, you saw it in the evolutionary history of plants, the maize had a polyploid ancestor. We were trying to trace it back to see why they are diploids now, and there's no wild polyploids around. Yeah.
Cool.
Sorry.
Maybe time for one more question.
Let's get on.
So our next speaker is Sire Kassama, who's been working in class on Bike's lab together with Eleanor Ritter, and she'll be telling us about proteins of obscure function in Arabidopsis thaliana, which sounds interesting to me at least.
All right, so today I'll be talking about proteins of obscure function that are found in Arabidopsis thaliana. And so I'll start off with some background, give an introduction to the plastoglobule, as well as an introduction to my target proteins, spread 1 and 2, as well as pigment 1 and 2. And I'll elaborate further on what those acronyms stand for. Then I'll give a brief overview of my methods, get into some results, conclusions, and future considerations. And so we're all fairly familiar with the chloroplast, pictured here. Has an outer membrane, inner membrane, thylakoid, stromal, granum. But what's not as well studied, but not as well known, but has been increasingly studied, is the plastoglobule. Often the PG. There's this yellow blob coming off the surface of the thylakoid. And you have the granule thylakoids, GT, and stromal thylakoids, ST. So plastoglobule is a lipoprotein that is found in all plastic types. And it has important roles in plastobiogenesis, homeostasis, senescence, as well as abiotic stress response. And a really good depiction of how plastoglobules respond to stress is this micrograph, the T and micrograph. And so this gray oval is the chloroplast. And you may be able to see some small black dots. Those are the plastoglobules. And you also have the thylakoids here. There's some sort of striations. And after 3 days of excess light stress, the plastoglobules increase in size, and the thylakoids kind of seem to disappear. This is of great interest to researchers. And so it has caused them to screen the plastoglobule for metabolites, lipids, as well as proteins. And they found something really interesting. The plastoglobule has a proteome of roughly 30 proteins, many of which have unknown domains and functions. Some have been studied though, and a co-expression network was developed. And so this looks like a lot, but there are some circles and some diamond shapes. So those are nodes, and each node represents one single gene. And then the diamonds in particular are plastoglobule genes. You also have lines. The gray lines are edges, and each edge represents a co-expression relationship. Red edges in particular represent co-expression relationships between plastoglobule genes. And so I really want to focus in on this boxed in portion because that's where my proteins are found. So we have flavonoid reductase related genes 1 and 2, and then we also have plastic-globule methyltransferase 1 and 2. And they have very tight co-expression with some other known proteins that work in cell death, seed metabolite content, seed longevity, and vitamin E synthesis. And so I want to step back and give a little background on flavonoid reductases. There are 3 classes of flavonoid reductases. And class 1 deals with the luminescence of bacteria, class 2 with flavin synthesis, and class 3. And this is where my friend proteins mostly fit into, depending on their domain homology, as well as sequence identity. This class deals with the synthesis of antibiotics, as well as the degradation of chelating agents. I also want to give some background on my plastic-globule methyltransferases. They have this particular domain, often denoted as SAM, and SAM methyltransferase is very broad, it's a large group of proteins, and these proteins are really interesting because their catalytic site can work with DNA, RNA, as well as proteins. And here, they have important roles in epigenetic regulation, as well as other really important processes. But pigments, my pigment 1 and 2 in particular, they have some interesting sequence identity to E. coli's ubiquinone methyltransferase. But surprise, ubiquinone isn't found in the chloroplast. So what are these proteins doing? What are pigment 1 and 2 doing, and Fred 1 and 2 doing? So that leads me to my objectives. So I'm going to stress phenotype my plants and look at mutant lines that have inactive genes for my proteins. And I'm also going to characterize the roles of Fred 1 and 2 as well as pigment 1 and 2. And so when we look at the molecular level of how a plant responds to stress, we can have a broader impact and see how plant breeders can develop cultivars that are better acclimated to dealing with our changing climate, as well as soil nutrition depleting in soil, the depletion of nutrients in soil. So that leads me to my methods. So I'm going to genotype all my plants, check for RNA expression of the genes, and then also look at some stress assays before those because that's what the plastic-globule responds to. Then I'll also look at data and collect data at root phenotype as well as chlorophyll concentration. And so I previously said that the plastic-globule responds to stress, and research shows that my target proteins respond to oxidative and salinity stress. And I'll just focus on oxidative stress right now. So I had a treatment group of 15 millimolar concentration of hydrogen peroxide. And you may also note that there's a pigment 1-2 cross. So we developed a double mutant of pigment because the pigment 1 and pigment 2 single mutants didn't really confer any type of phenotype. So from here on out, I'll just refer to it as pigment 1-2 cross. And when we look at the plants, they all look fairly similar. But when we look at the data, we see something interesting happen here. We have a wild type. It increases chlorophyll concentration upon induction of oxidative stress. But the pigment double mutant, it actually has a decrease in the chlorophyll concentration. This is interesting, and we see a similar trend with the Fred proteins as well. So the plants, they're in similar developmental stages, and they look pretty much similar. But the wild type, increasing chlorophyll concentration, and then a drastic decrease in chlorophyll concentration with the Fred 1 and Fred 2 proteins. The Fred 1 actually had a really high chlorophyll concentration in the control group as well. And so I also collected data on the root length as well. And I scanned all of my roots into WinRhizome software and measured root length, root surface area, as well as root diameter. The root diameter didn't really confer any type of phenotype. It was very similar across all of the plants. And root surface area and root length, it was really similar. And so I'll only be showing the root length data here today. And so we're looking at the salinity stress plants this time. And I had 2 treatment groups, one that was treated with 50 millimolar concentration of salt, and one with 75 millimolar treatment concentration of salt. And when we look at the wild type plants, the length increases in the 50 millimolar treatment group and then decreases at the 75. And then there's a slow, steady increase in root length as the concentration of chlorophyll increases. Concentration of salt, excuse me. And then the similar thing sort of happens with Fred, except it's a little bit more drastic in the 75 millimolar treatment group. And the Fred 2 mutant, it kind of does something interesting. I'm not really sure why precisely, but it starts off with this really long root in the control group. It decreases upon the 50 millimolar treatment group and then increases to a similar length as the control group was. So I keep thinking to myself, what's going on with these proteins? What methods and pathways are they affecting? And so I was able to develop a phylogenetic tree. Now, this also looks like it can be a lot, but I think it needs to be broken down. We're going to be mostly looking at the dicots, which are pictured in red, as well as yeast and fungi that are pink, cyanobacteria is blue, and then we have non-photosynthetic bacteria in orange. And so this phylogenetic tree gives us two important factors that were important to my study: the evolutionary history of my proteins, as well as possible functions of the proteins. And so if you look at Fred and 1 and Fred 2 genes, they're in two separate clades. They're possibly really, really far apart, and that's what I found out. Fred 1 appears to be your run-of-the-mill flower reductase that has been passed through and conserved throughout evolutionary history. But Fred 2 is a little bit more interesting. You see all this blue right here, that's all the cyanobacteria. And I postulated that the Fred 2 was introduced into the plants by an endosymbiotic relationship with cyanobacteria. And in particular, we have a bacillus and a streptomyces that have well been studied, flower reductases, that have well been studied. And these two proteins are actually homodimers that are involved in the synthesis of antibiotics. It's really important because it's possible that this Fred 2 gene works in a similar manner in some other pathway, but we're not really sure about that. What we do know is that they do have some common ancestors down here of E. coli. Now, these are mitochondrial transcripts, and so it's a little bit different. But we know that mitochondria and chloroplasts have similar evolutionary histories as well. They were introduced through endosymbiotic relationships. So this kind of concludes my talk for today, but I'd like to go over a little bit of what I talked about. We know that Fred mutants, well, the Fred proteins as well as the pigment proteins do confer phenotypes, a root phenotype in particular, and they both affect the chlorophyll concentration. And they may be involved in metabolic pathways. We're not sure which ones, but what we can do to figure this out is to express the proteins in the mitochondria of E. coli and see if they function in similar pathways. And then we could also screen the plasma globule for metabolites to see if there are some precursors in different metabolic pathways that are not being produced or just really in excess. And I'd also really love to acknowledge the Bandwidth Lab, especially my lovely mentor, Elena Michael. I'd also like to acknowledge the funding for this project, the National Science Foundation, some previous lab members for their development of the co-expression networks and some methods that has been established by project direction, Peter Lundquist and Lisa Giacomelli, the Bauer Lab, Juan Nunez, as well as Marie Svetsky for allowing me to use their root software, root scanning software, as well as some other people, Joseph Camerata, who helped me develop my phylogenetic trees, Delaney Sickler and George Daniel for putting this program together, as well as BT Guy and the Cornell University. Thank you. Are there any questions? Yes.
So in your co-expression graph, do the lengths of the edges correspond to the degree of co-expression?
No. Yes.
I'm wondering about the levels of your proteins in terms of oxidative stress. It's called oxidative stress. Do you know in terms of like gene expression levels or more in terms of protein levels?
No, you could also do some spectral counting to figure that out, but I wasn't sure.
And secondly, do you know if they're also kind of outside the plastic globule or are they specific?
They're specific to the plastic globule. Yes.
How do you know, like, to conduct the root assay? Was there something specific that?
Yeah, there was previous research that showed that the proteins they changed upon oxidative and salinity stress in the roots specifically. So that's what I meant.
Oh. Any more questions? Okay.
So on the phylogenetic tree, you say you have the gene duplication in the track one.
Yes.
Is that right?
Yes.
Is that localizing in TG as well?
Yes. Actually, no, it's in the chloroplast, just in the chloroplast in general, and it's not actually in the plastic globule. But there's also homologs to the Fred2 gene that are also found in the plastic globule, in the chloroplast as well.
That's CRISPR.
You make me so happy. Your love for CRISPR is pretty exciting. It makes me really happy. Don't ever lose your drive.
That would happen.
You're just really fascinated about CRISPR. When new technology is changing.
So our final speaker this morning's session is Natalie Gonzalez. She's in David Stern's lab, working with Amarillo, and will be telling us about targeting Cas9 to the chloroplast.
So, as George mentioned, my name is Natalie Gonzalez, and today I'm going to be presenting to you about my summer research, which was in determining the successful targeting of the Cas9 protein to the chloroplast. Since my project involves me working with chloroplasts so much, I'm actually going to begin by telling you a little bit of information about the chloroplast origin and how that information relates to the goals of the Stern Lab. Following this, I'll explain to you a bit about the CRISPR-Cas9 system and what it is, along with the constructs that the Stern Lab currently has available. I'll then follow this up with what my summer objective was, what methods I used to achieve that, and I'll follow that with some preliminary results and conclusions. So, if you're familiar with the theory of endosymbiosis, then you may already know that chloroplasts actually descended from a lineage of photosynthetic bacteria that are actually ancestral to modern-day cyanobacteria. Since the time of that endosymbiotic event, most of the chloroplast DNA has actually been transferred to the nucleus. That being said, the chloroplast still has the ability to produce some proteins that are important for photosynthesis, such as the large Nabisco subunit. Even still, most of the chloroplast proteins are now encoded for by the nucleus, and those proteins are targeted to the chloroplast through the use of the transit peptide. Now, I mentioned that the chloroplast genome has been reduced, but there have actually been some genes that have been newly discovered whose functions we don't totally understand. And it's the focus of the Stern Lab to actually better understand the function of those genes. So gene knockouts are a technique that's commonly used to better understand protein function. The problem is that up until this point in time, there have only been 2 organisms whose chloroplasts have been modified routinely. These include Chlamydomonas, who's cute, and Nicotiana tabacum, also known as tobacco. In contrast, there's a vast number of plants that you can do nuclear transformations with. So this setback presents the need for new tools that we can use to modify the chloroplast genome. In hopes of overcoming this obstacle, the Stern Lab has turned to the CRISPR-Cas9 system as a potential chloroplast gene editing tool. Now, the CRISPR-Cas9 system was originally discovered in bacterial cells as an immune response to viral DNA inserts. The CRISPR-Cas9 system is composed of a Cas9 endonuclease protein and a guide RNA sequence that is complementary to some specific sequence in DNA. So, this entire complex then goes along DNA molecules until it defines a sequence complementary to that of the guide RNA, at which point our Cas9 endonuclease will cleave out that region of the DNA in a double-stranded break. That break is then repaired by the host DNA repair machinery, resulting in indels. Now, going back to the goal of the Stern Lab, which is to better understand the function of those chloroplast genes, it's possible that we can use this Cas9 system to perform gene knockouts of those genes in order to test their function. The question then becomes, how do we go about using this system in chloroplast? Well, first, we would have to get both components of the CRISPR-Cas9 system into our chloroplast. So the Stern Lab has actually designed these 2 vectors here, and through the help of the VTI transformation facility, they've transformed the nuclear DNA of tobacco plants to produce either of these 2 constructs. The first construct here is of the Cas9 endonuclease, which is fused to a transit peptide sequence that will target that protein to the chloroplast. The second construct that I have here is of a guide RNA matching the large rubisco, sorry, it's complementary to the large rubisco subunit. And so if you're interested in learning more about this guide RNA construct, Leo Song, the other STERN Lab intern, is actually presenting a poster on this information. So this guide RNA construct has also been targeted to the chloroplast. So once we've obtained both these single lines, the TP-Cas9 line and the guide RNA RBCL line, crosses were formed between these lines to produce lines that contain both our constructs. One of the resulting genotypes of these lines produced pale plants. And this is promising because this may actually be evidence of a gene knockout of that large rubisco subunit. One step to confirming that the Cas9 system is in our chloroplast would be to confirm that both the CRISPR-Cas9 endonuclease and the guide RNA are in our chloroplast. So my part in this research this summer was confirming the presence of the Cas9 protein in the chloroplast. In order to do this, I had to isolate chloroplasts, at which point I did protein extractions with them. I also collected proteins from total plant tissue because I wanted to be able to do a comparison between my plant proteins and the chloroplast proteins. Because the Cas9 endonuclease is being targeted to the chloroplast, I expected to see an enrichment of that protein in the chloroplast. Now, in order to visualize this, I loaded my proteins into an SDS page gel and did an immunoblot with those using an antibody specific for the Cas9 protein. So for my chloroplast isolation, I collected seedling tissue, at which point I blended those up and created this homogenate here. I then filtered that through several layers of cheesecloth, which then I pelleted down that homogenate to get this chloroplast pellet here, which I then transferred to a Percol gradient. Those Percol gradients were then spun down, and through that process, intact chloroplasts migrate through the Percol gradient to about there. Then I collected those intact chloroplasts and stored them at -80 for later use. For my protein extractions, I placed either plant tissue or chloroplast isolates into liquid nitrogen, at which point I proceeded to pulverizing my samples. I then added protein extraction buffer to those samples, spun them down, and collected the supernatant, which contains my proteins. I then used those proteins in SDS page gels to perform immunoblots. So shown here is actually one of my first SDS page gels that I stained with Kumasi. This gel is loaded with total plant proteins, and my first lane contains a negative control that I've acquired through the Joyce Van Eyck's lab, as well as a positive control, which I also acquired through the Joyce Van Eyck lab. The following lanes are of our TP-Cas9 only plant extractions, our guide RNA only plant extractions, and our TP-Cas9 guide RNA cross that should contain both our constructs. Now, through the use of the Kumasi stain gel, we can evaluate protein loading and integrity. For example, at the 50 kilodalton size, we see this consistent band across all our lanes, which implies that similar amounts of protein were likely loaded across all our lanes. If we saw smearing of these bands or lanes, then this would be indicative of protein degradation. So those same samples were then loaded into a 12% SDS page gel that I then did a semi-dry transfer with onto a PVDF membrane, and I used a transfer buffer that contained 20% methanol through this process. So the Cas9 protein is supposed to migrate at about 158 kilodaltons, and with that transit peptide fused to it, it's supposed to migrate at about 167 kilodaltons. The lack of a band at either of those sizes indicates that we do not have Cas9 protein on our membrane. And this may have been due to issues with the immunoblot protocol. So we decided to optimize this protocol in order to transfer larger proteins. So we first did this by switching to a wet transfer, which is said to be better for transferring larger proteins onto the PVDF membrane. We also removed the methanol from our transfer buffer because it said that the methanol and the SDS can react in a way that cause our proteins to precipitate, and that would hinder the ability of that protein to be transferred onto the membrane. Lastly, we switched to an 8% gel as opposed to a 12% gel in order to get better separation of our larger proteins. Okay, so using this modified immunoblot protocol, we were actually able to detect the Cas9 protein through the use of the Cas9 antibody. And in this case, prior to loading my samples, I performed a Bradford assay to quantify the amount of protein in my samples. My goal was to equally load proteins across my lanes. So recall that the Cas9 protein is supposed to come in at about 160 kilodaltons, which is what we see here in our Cas9 standard. The next sample is loaded with total protein extraction and chloroplast protein extraction from our guide RNA line, followed by a TP Cas9 line, and then 2 different crosses between those lines. You can see that for our total protein extraction of the guide RNA line and for the chloroplast protein extraction of our guide RNA line, there's no band at that 160 kilodalton size, which indicates that there's no Cas9 protein in that guide RNA line. And this is expected because the guide RNA line does not contain our Cas9 construct. The next set of samples is from our TP Cas9 lines, and again we see that in our total protein extractions, there's no band at the Cas9 size. However, in the chloroplast extraction, we see that 160 kilodalton size, indicating the presence of the Cas9 protein. And we see that trend in our crosses as well, where we have no protein band at the 160 mark for the total protein extraction, but at the chloroplast lanes, we do have the 160 kilodalton band. So I then loaded those same samples because through this process, we actually can't confirm that we have enrichment in the chloroplast of that Cas9 protein. So I loaded those same samples into another SDS page gel that I did a Kumasi stain with. And you can see that these wells were not equally loaded across with proteins. And in fact, some of our chloroplast extraction lanes are overloaded, while some of the total protein lanes are underloaded. And furthermore, the large Rubisco subunit band right here in our TP Cas9 line is way more overrepresented than that of the chloroplast. So to conclude, we were finally able to confirm that the Cas9 protein is actually in the chloroplast of our TP Cas9 constructs. Moving forward with this project, it would be a good idea to be able to determine whether or not there is an enrichment of that protein in our chloroplast. And we can do that by repeating the results of the last gel, but ensuring that we load equal amounts of protein in each of our lanes, which we can quantify through the use of a Bradford assay. Furthermore, we would be able to, if we used different antibodies, it would help us further establish whether or not we have the enrichment of the Cas9 protein in our chloroplast. So for example, if we use chloroplast antibodies specific to chloroplast or chloroplast targeted proteins, and our protein localization pattern of our Cas9 gels matched that of the chloroplast bands and the chloroplast targeted bands, then it would be a strong indication that those bands are actually, that the Cas9 protein is actually enriched in our chloroplast samples. In order to confirm that we have isolated chloroplast, we could also use a cytosolic antibody as a control for that. Lastly, I understand that the Stern Lab has transformed some Arabidopsis plants in similar ways to contain these constructs. So a next step would be to confirm that the Cas9 system is also getting into the chloroplast of the Arabidopsis, which is something that's never been studied before. So I'd like to thank the Storm Lab, first of all, my PI David Stern, and my mentor Amber Poto, who has been an absolute pleasure to work with this summer. I would also like to thank the BTI Transformation Facility for providing some of our control samples, as well as Delaney and George for their coordination of the program, and my funders, the NSF program and the Triathlon Foundation. So, questions?
Did you get the opportunity to look at or help with the synthesis of guide RNAs?
With guide RNAs? No, I didn't actually, but Leo was the one that actually worked with that guide RNA or ECL construct, so he probably knows a lot more about that. Yes?
So the new protocol to use for the detection of Cas9 in gel was amazing. How do you know which parameters to set? You try one based on multiples?
So, to be honest, most of these results I got within like the last couple of days. So most of the summer has been me troubleshooting that, and we tried this by loading different amounts of protein into our samples. You can see with like the RBCL line, we had overexpression. And if you do an immunoblot with that, the bands come out pretty fuzzy, so you need to make sure that you have the right amount of samples, the right amount of antibody and everything. So it was just a lot of troubleshooting. Yes.
Going along with the new protocol, how could you be sure that the Cas9 still moved? I think it was TB Cas9, it still was at 158 kilodalton. Since you used a new protocol, how were you sure those are the same?
So we load actually a protein ladder, which I didn't include here, that's the markers were on the side. And so if you run the protein ladder with your proteins, you can see where matching bands line up, and you'll say, okay, well, this band lines up at about the 50 kilodalton mark. So that's got to be about 50 kilodaltons more or less. Did that answer your question? Yes. Okay, perfect. Yes?
The Cas9 protein is a rather large protein. Do you know if the size affects the efficiency of incorporation into the chloroplast?
Yeah, so that's actually the whole problem that we had with the original gel. We weren't getting that transfer, and so initially we thought maybe that Cas9 protein just isn't in our samples for some reason. But then actually someone in the lab had suggested that a couple of papers that they had read mentioned using that wet transfer process for larger proteins.
I'm not talking about in the gel, I meant into the actual chloroplast. So you have the transit peptide on the protein. Do you know if the size of the actual protein is impacting potentially the way it's incorporated efficiently into the chloroplast?
I don't actually know that, but as I mentioned earlier, the chloroplast, most of the proteins are encoded for outside of the nucleus. So I'm sure there's like a range of large proteins that also manage to get in there.
Okay, well, and that's thanks all of our speakers. Things. The next on the schedule is lunch and poster session. The poster presenters need to get to their posters, so please, if you're not a poster presenter, stick to the back. And if you are a poster presenter, feel free to cut in line and get in early so that you can see
Yeah, the protein average 75 kilodalton, so that's a big protein.
I don't know.
They're all equal. I just look at these.
talk to judge.
Something like that. So,
I hope I answered it.
yeah, no, I was just wondering in terms of this.
What's really interesting is that some established schools, so what is that, things like after being Western, use light on chairs in a way, like overall. And in some, most times, yes.
Which is sort of theoretically what you would expect because in most cases the problem is solved.
Right, like in some, well, what's really interesting is if you look at sort of history, because in the established colleges, even though you see like Francis and settled down, you can see sort of the ancient patterns of the mess in like all the chromosomal rearrangement stuff.
Yeah.
But in some colleges, you don't see that at all. Has virtually no, but you can still, even though it's like a really old organization, you can still see perfectly the ancient stuff.
Right.
And so how, but when you resynthesize it,
Inferring,
it's a
including
different
through
lens.
therefore. Okay. So that's
So
great.
how-
That's useful. Also, unless it was like a really special. Which sort of highlights hazards of inferring like old ones are just like the same ones that we're doing now.
Well, and the fact is really interesting what's cool about Carla Dizon is that the heterozygous one is virtually, the non was virtually the same as the difference, whereas the homozygous one is much lower, which you might expect. But so there is a problem there. Yeah, but in the heterozygous ones, when I look at spreads, you can see completely non-homologous, not even non-homologous, completely non-homologous, like chromosome 1 and chromosome 4. And so, how is that happening? And yet it still has an 80%